Assalamu alaikum. This is Professor Ma'moon Ahram. I'll be with you in this part of the cytology and molecular biology course. I'll be covering the molecular biology part of the course. So let's start. First of all, a really important note is that the main source of the lectures is the lectures themselves. Okay, so there is a textbook. It's a beautiful, excellent textbook called the Cell A Molecular Approach. It is available free online. Actually, it has been hacked. You can download it. So it's really an excellent textbook. I love the figures. The figure legends are detailed, so you can read them, and you don't have to read, for the most part, you don't have to read the whole book. But the main resource is the lectures themselves, okay? So I'm not gonna ask you questions from the textbook itself, rather just from the lectures. So these are the different topics that we'll be covering in this course. We'll start with nucleic acid structure. So this will take 2 lectures. Some lectures will be, or some topics will be covered in 1 lecture. Other topics will need 3 lectures. So we'll start with the nucleic acid structure. Then we'll follow this up with basic techniques. Then we'll have a lecture on restriction in the nucleases, also related to techniques as well. So then we'll cover the human genome, DNA replication, transcription, regulation of transcription in both prokaryotes and eukaryotes. Then we'll have a lecture on analysis of gene expression, that is gene activity, and this is another technical lecture. And then we'll have 3 lectures on translation and its regulation, and then we'll end up with DNA mutations and DNA repair. So let's start with the first lecture. Again, the main resource for this lecture is the lecture itself, whatever I cover. And you can refer to Cooper's chapters 2, 4, and 6, and there are different pages that are scattered. You can always refer to the index at the end, and you can just look up different keywords that are related to the lectures. And you can go back to these pages. So hopefully, I hope that the number of pages in the textbook is accurate. But again, just refer to the index. Okay, let's start. So we'll start with what molecular biology is, and the main concepts, or the main philosophy, let's say, of molecular biology. So molecular biology really is biochemistry. So it talks about the different reactions, the biochemical structures, the information in these molecules, but it's specifically related to DNA and RNA, okay? So it's a branch of molecular biology. It is not genetics, okay? There's an overlap between genetics and molecular biology. Genetics deals with patterns of inheritance of phenotypes and genotypes, or the genetic material and the physical appearance and characteristics of individuals. It deals with inheritance, it deals with heredity, it deals with chromosomal structures rather than small molecules like DNA and RNA. Okay, so molecular biology deals with biochemical reactions that take place in DNA and RNA. Now, there's something known as the central dogma of molecular biology, and the central dogma of molecular biology is basically the following. You have a DNA molecule, and this DNA molecule is the genetic material of cells or organisms, and this DNA molecule is used to make an RNA molecule, and this is done via a process known as transcription. Okay, now and it's catalyzed by an enzyme known as RNA polymerase. Now, RNA is then used to synthesize proteins in a process known as translation, and this involves ribosomes. Okay, and DNA can make a copy of itself via a process known as replication. So, and it's catalyzed by DNA polymerase. So this is really the central dogma of molecular biology, which is basically DNA is used to make RNA and RNA is used to make proteins. That's it. Now, of course, things have become more complex, okay? For example, you can make DNA out of RNA. You can make RNA out of RNA, and so on. So things are getting more complex as we understand more and more of how nature functions and works. So in this course, we'll talk about these processes. As I said, we'll talk about the structures of DNA and RNA. We'll talk about the transcription and translation. We'll talk about replication as well, that is making DNA out of DNA. Okay. Now, we'll also talk about the organization of our DNA. We'll talk about mutations and repair. We'll talk about the different types of RNA molecules. Okay. We'll talk about techniques that involve replication and transcription throughout this course. But central dogma of molecular biology is DNA is used to make RNA, RNA is used to make proteins, and DNA can be used to make another copy of DNA. Okay? Now, what are nucleic acids? There are two types of nucleic acids. One, you have DNA, which is the abbreviation for deoxyribonucleic acid. Okay? And you have RNA, and RNA stands for ribonucleic acid. So we'll go over the differences between DNA and RNA molecules. So basically, the primary structure of a DNA molecule or a nucleic acid is basically you have nucleotides. So these are known as monomers, okay? And they are connected to each other to form this large polymer, DNA polymer in this case. Okay, so what a polymer is basically it's a molecule that is made of repetitive units. In this case, it's nucleotides. Okay, now these are organized in a larger structure known as chromosomes. So what are nucleotides? A nucleotide is a subunit of nucleic acids. It is composed of three components. Okay, the one component is a sugar, and this sugar is a 5-carbon sugar. It's a pentose. So penta means 5. So if I say, for example, di, it means 2. Mono means 1. Tri means 3, then you have tetra means 4, and then you have penta. Okay, so so this is a 5-carbon sugar, carbon 1, 2, 3, 4, and that's number 5. And the sugar is different in RNA versus DNA. So in RNA it is a ribose. So ribose is a 5-carbon sugar, okay? So it is a ribose. This is the sugar that exists in RNA, ribonucleic acid. So that's ribose, okay? In DNA, it is a deoxyribose. Deoxy means that it lacks an oxygen, and this oxygen is right here. It's associated with carbon number 2, right here. Okay, so in RNA it is a hydroxyl group, but in DNA it's just a hydrogen. Okay, so it is a deoxyribose in DNA, and DNA stands for deoxyribonucleic acid. Okay, now that's one. Now the other component is a phosphate group. Okay, so and we'll talk about this phosphate group in a second. So there's a phosphate group, and note that this phosphate group has negative charges associated with it, and that what makes DNA and RNA acidic molecule. So deoxyribonucleic acid or ribonucleic acid. And the third component is basically a nitrogenous base. Okay, so we'll talk about this base right here, and there are 5 bases that are associated with the ribose. Now note that the bases are associated with carbon number 2. Now another important carbon is carbon number 3, and another one actually is carbon number 5. So these are really important for you to know. So what does this mark mean? This little thing right here. This is known as prime. So this is 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. So why do we have this? Well, to differentiate the carbons in the sugar versus the carbons in the bases. So if I say carbon number 1, here I'm talking about the carbon that exists in the bases, or carbon number 2. Okay. Now if I say carbon 1 prime, here or the 1 prime carbon, here I'm talking about the carbon in the sugar itself. Okay, so that basically what it means. Now this is biochemistry right here. The association, the bond between carbon number 1 in the sugar and the group in the nitrogenous base is known as a glycosidic bond. So let's talk about the nitrogenous bases now. As I said, there are 2 types of bases. You have what is known as pyrimidines and you have purines. Now notice the numbering of the different groups right here. So that's group number 1, that's a nitrogen, carbon 2, nitrogen 3, carbon 4, and so on. You don't have, by the way, to memorize this, but this is just to show you that we're using the word prime to symbolize the different groups of the ribose molecule. Okay, to differentiate the groups of the ribose from the groups in the purines and pyrimidines. Okay, so to nitrogen number 1, you have the glycosidic bond between the base and the sugar. And to nitrogen number 9, you can also have the formation of the glycosidic bond between the purine and the sugar. Now notice that pyrimidines are single ring molecules and purines are double ring molecules. So how can I differentiate between the two? Well, that's how I learned it when I was a student. The little word is for the big structure, and the big word is for the small structure. Okay, so pyrimidine is for the single ring molecule and purine is for the double ring molecule. Now there are different types of purines and pyrimidines. Okay, so if I say pyrimidine, I'm talking about this structure, but this structure can have different forms. So for pyrimidines, there are 3 different types: cytosines, thymines, and uracil. Okay, now notice the differences between them. So for cytosine, for example, you have a ketone group right here, right, and you have an amino group in here. Now for thymine, it looks exactly like cytosine, except that there is no more amino group, there is rather a ketone group right here. Okay, now uracil looks like and thymine also has a methyl group. Now uracil looks like cytosine, except that it has cytosine has an amino group and uracil has the ketone group. Okay, now you will learn later on that cytosine exists in both DNA and RNA. Thymine exists in DNA, but not RNA, that's in humans. Okay, it exists in some RNA molecules in like bacteria and so on. But we're mainly focusing on human cells. And uracil exists only in RNA molecules. Okay, so there are differences between DNA and RNA, and throughout this lecture we will talk about these differences. Okay, so you need to pay attention to that. Now, purines, the double ring molecules, there are two types that make up nucleic acids. You have adenine and you have guanine. Now, a major difference between them is that for adenine, you have an amino group right here. For guanine, you have a ketone group right there. Okay, so now there are different types of purines and pyrimidines, by the way, so it's not only these five, but these are the 5 molecules that make up nucleic acids. These are the main ones that we have to focus on. So as I said, there are differences between DNA and RNA. You will learn that DNA is mainly double-stranded, RNA is mainly single-stranded, so it's made of one strand. That's for RNA, and DNA is made of 2 strands. For DNA, it's a deoxyribose. There is a hydrogen. For RNA, it's a ribose. There's a hydroxyl group. Now, in terms of the bases, they both contain cytosine, adenine, and guanine, but DNA mainly contains thymine, and uracil exists in RNA only. Okay. Now, there are always exceptions, especially in lower organisms like viruses and bacteria. Okay, so let's learn a few terms in here. I said that nucleotides make up DNA and RNA. So what is a nucleotide? A nucleotide is basically a molecule that is composed of 3 components, and these are the sugar, the pentose sugar, the nitrogenous base, and number 3, the phosphate. Okay? Now, if you remove the phosphate, you will end up with 2 components, and these are the sugar and the base. Now, this molecule is known as a nucleoside. So a nucleoside is basically a sugar with a nitrogenous base linked by a glycosidic bond. If you add a phosphate group, then it is known as a nucleotide. Now, a nucleotide can have actually 1 phosphate group, 2 phosphate groups, or 3. So if I say this is a nucleotide, I would know that it contains a sugar, a base, and a phosphate. But how many phosphate molecules? I don't know. It could be 1, 2, or 3. Okay? So in order to be very specific with the naming, we say it's a nucleoside monophosphate. You see? So if I say nucleoside monophosphate, it's a nucleoside, so it contains a sugar and a base. And if I say monophosphate, I know now it's a nucleotide and it has 1 phosphate group. Okay? So I can be more specific and say nucleoside diphosphate. So I know it's a nucleotide because it contains all 3 components, and specifically it has 2 phosphate groups. Similarly, I can say nucleoside triphosphate, so it contains 3 phosphate groups. Okay, so this naming is really important. So let's look at naming again. So you look at this molecule, for example, and you notice that, whoa, it contains a sugar, a base, and a phosphate group. So you know it's a nucleotide. So what nucleotide exactly? Okay, you look at the sugar. So okay, so that's number 1. You look at if the 3 components exist or not. Okay, if they do, you know it's a nucleotide. So you look at the phosphate. How many phosphates? Is it 1, 2, or 3? If it is 1, you know it is a monophosphate. Okay, if it's 2, then you know it's a diphosphate. 3, triphosphate. Then you look at the sugar and look at carbon number 2 right here. it has a hydroxyl group, right? So you know it is a ribose. It is not a deoxyribose. This here is a deoxyribose. Then you look at the base. Is it a purine or a pyrimidine? So you notice that it is a purine. Again, the small word for the big structure. So it's a purine. Great. Then you look at specifically what it is exactly. You notice that there's an amino group, so you know it is an adenine. Okay, great. Now, as a nucleoside, that is, if you remove the phosphate, it becomes adenosine. So adenine is the base. If it is linked to a sugar, it becomes adenosine. Okay? Now, if you add a phosphate to it, it's a nucleotide and it's only one phosphate, so you say it is adenosine monophosphate because you have one phosphate group. It is abbreviated as AMP, okay, or simply just an A. Now, if I say adenylate, adenylate means it's a simplified term for adenosine monophosphate. Okay, just say adenylate. Now, let's look at the other one right here. So again, you look at it, you say, you see that there is a sugar, a base, and a phosphate, so it's a nucleotide. Great, how many phosphates? Just one phosphate, so it's a monophosphate. Is it deoxyribose or ribose? No, it is a ribose molecule. Great. So you look at the base, is it purine or pyrimidine? It's a purine. Great, you look at it specifically and you say, aha, it's a guanine. So as a nucleoside, you say it is a guanosine. When you add a phosphate, it becomes guanosine monophosphate. So that's the name of the nucleotide, or you can simply say guanylate. Now you look at the other two. Again, same thing. It's a nucleotide, monophosphate. It's a ribose, not a deoxyribose. You look at the base. It's a pyrimidine. Specifically, it is a uracil. Okay. So it's known as uridine monophosphate or uridylate. For cytosine, if it becomes part of a nucleotide, then it becomes cytidylate or cytidine monophosphate. Okay, and note the abbreviations CMP, C, UMP, U, and so on. So these are the nucleotides that make up DNA. So you have deoxyadenosine or deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxythymidine monophosphate or deoxythymidylate, and you have deoxycytidine monophosphate or deoxycytidylate. Okay, again, note the abbreviations. D stands for deoxy, so DA, deoxyadenylate, and so on. Deoxy-AMP, deoxy-GMP, deoxy-TMP, deoxy-CMP. Of course, if you have two phosphates, for example, in here, it becomes deoxy-ADP. If it contains three phosphates, it becomes deoxy-ATP. Same thing here, ATP and ADP and so on. Okay. Now, to make up DNA and RNA molecules, you have to link the nucleotides to each other. And these nucleotides are linked to each other via a bond known as a phosphodiester bond. Okay, so right here, this is known as the phosphodiester bond. Di means two, by the way, because right here you have two phosphoester groups, 1 and 2. So note that the link occurs between carbon number 3 with a phosphate that is linked to carbon number 5. Okay, so pay attention to that. We'll talk about this in a second. Okay, so then you can have another nucleotide that is added right here. You can have the phosphate group of the other nucleotide added to to carbon number 3 of this sugar molecule. So you can elongate, you can make the nucleic acid longer in this case, and so on. Now notice the bases. Here you have a cytosine, here you have a guanine, and so on. So this is how nucleic acids are formed. Okay, so RNA and DNA molecules are polymers. And polymers, as I said, these are molecules that are made of repeated units of monomers. So a nucleotide is a monomer. So right here you have a monomer, and it's linked to another subunit, as we call it. Okay, so unit is one, subunit means that it's part of a larger structure. So you have another subunit right here, you have a third subunit right there, and so on. And note the link, the phosphodiester bonds between the different nucleotides. Now if I ask you, is this a DNA molecule or an RNA molecule? Now you can tell by two things. Primarily, you look at the sugar molecule, and you look here, carbon number 2, it doesn't have a hydroxyl group or just a hydrogen, no, it contains a hydroxyl group, then you say then it is an RNA molecule. The other hint is you look at the bases. Does it contain uracil? And aha, it does. It does contain uracil, so you say it is an RNA molecule. Does that mean that uracil does not exist in DNA? You know what? It does, and when it does, it's a mutation. It is something that is wrong. Okay, it doesn't happen. It shouldn't happen in DNA. So it has to be repaired. It has to be removed and replaced by something else. Okay, so here you have a nucleic acid polymer, an RNA molecule. Now note this, the numbers right here. This here is known as the 5' end or the 5' terminus, and the other end is known as a 3' end or a 3' terminus. Okay, so it is a polar molecule, meaning that it has two opposite or two different ends. So if I tell you what is the order of bases of this RNA molecule, you have to say it is adenine, cytosine, guanine, uracil. Okay, you cannot say, or you can say it's A, C, G, U. Use the abbreviations. You never ever say it is U, G, C, A. You have to start with the 5' end all the time. Why? Because whenever we want to make this RNA molecule longer, we add the nucleotide to the 3' end. We never add it to the 5' end. So this is fixed. It doesn't change. So it gets longer from the other end, and that's why we have to say it is A, C, G, U, and whatever comes next until we get to the 3' end or the 3' terminus. So these ends, 3' and 5' are really important, okay, as a terminology. So let's look at this molecule right here, this polymer right here. Again, repeated units of nucleotides. So you look at the sugar, aha, it's a deoxy sugar. So you know it is a DNA molecule. The other hint is you look at the thymine and you say, aha, it is DNA. So if I say, what is the sequence? Pay attention to the wording. If I say, what is the sequence of the DNA molecule, what I'm talking about here, what I'm asking for right here is, what is the order of nucleotides in this DNA molecule? What is the sequence of the DNA molecule? And you say it is T, G, C, A. Now, if I don't know what this molecule is, I look at it and I say, aha, this is a deoxy T or DT, this is DG, this is DC, this is DA, and so on. But if I say this is a DNA molecule and its sequence is TGCA, you know that I'm talking about a deoxy nucleotide. I'm not talking about a regular nucleotide with a hydroxyl group right here. Okay? Now, so that's the sequence of the DNA. Same thing, you have 5' end and you have the 3' end, and you start reading the nucleotides from the 5' end, TGCA, because whenever we want to make this DNA molecule longer, we add the nucleotides to the 3' end. So this is fixed, it doesn't change, but the 3' end becomes different. Okay. Now, let's look at the DNA structure. The DNA molecule has a certain characteristics, and these are listed in here. Okay. So first of all, it's a double helical molecule. It's a double helix. Okay, so this is the famous name for the DNA molecule. It's a double helix molecule. What I mean by helix is that it looks like a spring, right? So it rotates, it keeps on rotating. And you have two strands, we call them two strands, that intertwine around each other. Okay, in a helical formation. So notice here how you have this strand in front, and then it rotates, it goes to the back, and this one comes here to the front, and so on. So it's a double helical molecule. Now, the DNA structure, by the way, was revealed by two scientists known as Watson, this is Watson, and Crick. This is Crick right here. And they did something known as crystallography. Basically, they converted DNA molecule into a crystal and they hit it with X-ray, and you got this pattern. And based on this, and I don't know how they did it, but they interpreted this image into this structure. But there is dirty history behind it, by the way. So anyhow, they got the Nobel Prize, I think, in 1961 or 1962, and their discovery was published in 1953. Really famous scientist. This person is still alive, Watson is still alive, and he got into trouble like 2-3 years ago with racist remarks. Anyhow, so DNA is a double helical molecule right here. That's how it looks like. Now, it is not a perfect helix. This what a perfect helix would look like. Okay, really uniform helical structure. Rather, you have it sort of like bent, as you notice in here. Okay, so it's not a perfect helix. So that's the first one. It is a double helical molecule. Now, there's a scientist known as Chargaff, I think he was Russian, and he noticed in the 1940s something that is really interesting about DNA molecules, and that is the number of A's is always equal to the number of T's, okay, as nucleotides, okay? And the number of G's is always equal to the number of C's. And the number of purines is always equal to the number of pyrimidines. Okay. Now, but it's not necessarily that the number of A's and T's would be equal to the numbers of G's and C's, by the way. Okay. So again, A is equal to T, G is equal to C, and purines are also always equal to pyrimidines. So based on this information, Watson and Crick came up with their model for DNA structure. Something else about DNA structure is that these two strands are linked to each other via hydrogen bonding between the bases. So whenever you have, for example, an A right here, so that's an A, it's always hydrogen bonded to a T. This T exists on the other strand. And the number of hydrogen bonds is always 2. Now, on the other hand, Ag is also hydrogen bonded to a C. And the number of hydrogen bonds is always 3. Okay, so you have two bonds, two hydrogen bonds between A and T, and you have 3 hydrogen bonds between G and C, and this has very important implications about the stability of the DNA molecule as a double-stranded molecule. Now, you don't have to memorize the pattern of bonding, by the way, or or how these bonds are formed between the different molecules. Okay. Now, as a result, we say that DNA is complementary, meaning that the bases complement each other, so that whenever you have a C, you have a G, okay, and whenever you have an A, you have a T. And the number of hydrogen bonds, note, the number of hydrogen bonds between C and G is 3, between A and T is 2. Great. So we say that the DNA molecule is a complement, the two strands are complementary to each other. They complete each other. Whenever you have a C, you have a G. Whenever you have an A, you have a T. Whenever you have a T on one strand, you have an A on the opposite strand. Whenever you have a G on one strand, you have a C on the other strand, and so on. So that's another characteristic of DNA. Another one is that if you look at the structure, by the way, there is what we say a backbone, and we have side chains. So the backbone basically is the straight line of phosphate, sugar, phosphate, sugar, phosphate, sugar. This is the backbone. Now the side chains are the bases themselves. Okay, they extend almost perpendicular to the backbone. Okay, so this is an important concept, by the way, in organic chemistry and in biochemistry as a whole. That is, you have, especially in polymers, for polymers you always have this backbone, and sometimes you have side chains as well. So when we talk about proteins in the biochemistry course, you would notice that there is a backbone and you have side chains as well. Okay. Now, something else about DNA is that it is anti-parallel. What do I mean by that? Remember, it's double-stranded molecule. Okay, and each one of these strands has a 5' end and a 3' end. Okay, but the two strands are not parallel to each other. They are in fact anti-parallel. What we mean is that you have one strand where on top you have 5' end. Okay, so that's the carbon number 5 associated with phosphate. Okay, and on the other end you have the 3' carbon right here. Okay, so that's the 3' end. Now if you look at the opposite, the complementary strand, you would notice that actually on top you have the 3' end, and in the bottom you have the 5' end. Okay, so the two strands are anti-parallel to each other, and this is another important characteristic of DNA. Now, whenever, by the way, we write the sequence of nucleic acids, we can do them in different formats. So for DNA, I can do it this way. For one strand, 5', A, T, G, G, C, C, etc. Okay. Now, I can write the opposite strand as well, and it has to be anti-parallel, and it has to be complementary as well. So you say 3 prime T A C C G. Okay, so whenever there's an A, there's a T. Whenever there's a T, there's an A. Whenever there's a G, there's a C. Whenever there's a C, there's a G, and so on. Okay, so the molecule is complementary, is double-stranded, and the two strands are complementary to each other, and they are anti-parallel to each other as well. Now, for RNA, since usually it's a single-stranded molecule, we say 5 prime A U. Aha, notice the U. There's a U. There's no G. G, G, C, C, U. Now, for DNA or RNA, by the way, I can say, for DNA, for example, I can say, if I ask you, what is the sequence of this DNA molecule, you can just say, it's A, T, G, G, C, C, T, G, whatever. Now, you don't have to tell me two things. One, you don't have to tell me 5 prime and 3 prime. Because once you start with A, T, G, G, I know that you are starting with you're starting at the 5 prime end, and you end at the 3 prime end. Okay. Number two, the second thing that you don't have to tell me is what the complementary strand is. You don't have to write this whole thing. If you just say it's A, T, G, G, C, C, I know that you're starting at the 5 prime end, and I know that the complementary strand is T, A, C, C, G, G, and I'm starting the complementary strand at the 3 prime end. Okay? Now, if I want to read just the complementary strand, I would have to do it from this end, by the way. From the 5 prime end, I would have to say T, G, A, unless I say 3 prime T, A, C, C, G, G, 5 prime, then I know exactly where I'm starting at. So if I don't say 5 prime and I start reading the bases, you should know that I'm starting at the 5 prime end. I hope it's clear. Same thing with RNA, by the way. Okay, so DNA is flexible. Okay, it's a stable molecule, but it's flexible, or I can say DNA is flexible yet stable. So basically the idea is that it's like an electric wire right here. Okay, if you try to stretch it, you try to rip it off, you wouldn't be able to. Okay, because it's really strong. It's a strong structure, just like DNA. Okay, but it's flexible just like an electric wire. It can be bent, and this is an important characteristic in DNA. This is how it is packed in our in the nucleus of our cell, of every cell in our body. This is how it interacts with proteins as well. Okay, because it's flexible, it can be bent. Now, so DNA has a lot of phosphate groups, and these phosphate groups carry a lot of negative charges. So on the surface of DNA, there are these phosphate groups that would cause repulsion, or they would result in repulsion because all of these negative charges. Okay, and that sort of destabilizes DNA a bit. Now, one characteristic of DNA is that as a result of having all of these negative charges, it attracts negatively, possibly charged ions like sodium ions and magnesium ions. So what these do is that they associate with these ions, okay, like stabilizing DNA. The other thing is that DNA also is attracted, or it attracts, it binds, it associates with proteins known as histones, and these histones carry a lot of possibly charged ions as well as a result of having these amino acids that, like lysines, specifically lysines, and that's how DNA is wrapped around histones. Something important about DNA molecule is that since it is not a perfect helical molecule, it contains what we call as grooves. Okay, so there are two grooves in here for a DNA molecule. You have what is known as a minor groove and you have a major groove right here. Okay, notice the space in the major groove. It's larger than the minor groove. Now, and they alternate. So you have a minor groove, major groove, minor groove, major groove, minor groove, major groove, and so on. Now, opposite to a minor groove, you have a major groove. And opposite to a major groove, you have a minor groove. So why is that important? So these grooves are important for facilitating the interaction between DNA and proteins. So this here is a DNA molecule. So notice here you have a major groove, and right here you have a minor groove right there. Okay, so you have a major groove in here and you have a minor groove in there, and so on. So proteins need space. So what happens is that proteins can insert themselves within the major groove, and the amino acids of proteins can interact with the exposed groups of the nucleotides. Okay? Notice that, for example, this part of the protein cannot insert itself in the minor groove, but these two parts can. Now, once they insert themselves into the major groove or grooves, they would be closer to the nucleotides and they can interact with these nucleotides. And that is why proteins can interact specifically with specific sequences of DNA, because you have specific amino acids within the protein that exist in a certain space that can interact with specific sequences or specific nucleotides within the major groove. Okay, and this specific interaction is very important as well when we start talking about replication and transcription and so on. So the importance of the major groove is that they facilitate interaction with proteins. Okay. Now, so again, you have this interaction right here. That's in case I flip it. You have the bases, by the way, are not totally embedded within the strands. Rather, you have some groups of the nucleotides exposed outside the nucleotides, and these groups can interact with amino acids within the proteins. Okay, so you have this specific interaction between DNA and proteins. And something else that I should emphasize is that what's important about biochemistry or molecular biology is really not necessarily the covalent ones. It's rather the non-covalent interactions. Okay, so whenever we're going to talk about different processes, molecular processes that take place in our cells, or different reactions that happen in our cells, they are mainly specified by non-covalent interactions. And here we're talking about 4 types of interactions. We're talking about hydrogen bonds, electrostatic interactions, hydrophobic interactions, and finally van der Waals interactions, or London dispersion interactions or forces. Okay. Now, there are differences in the way chromosomes look like in prokaryotes and eukaryotes. You know that eukaryotes. Eu means true, meaning that it's a cell that contains a true nucleus. Prokaryotes meaning that they lack nuclei. So you have DNA swimming sort of in the cytoplasm, whereas in eukaryotic cells you have DNA enclosed in a nucleus, surrounded by a nuclear membrane. Now, this is one difference. Another difference is that in prokaryotic cells, in general, you have DNA organized as a single chromosomal molecule that is round or circular chromosome, whereas for eukaryotic cells, cells contain multiple chromosomes, and these chromosomes are linear rather than circular. So in human cells, we have our chromosomes, our DNA organized as chromosomes, and these chromosomes are linear and they are enclosed inside nuclei. Okay. Now, the thing is, how is DNA organized in eukaryotic cells? Because if you look at the DNA molecule in a single cell, it's about 2 meters long, so it's really large, okay, 2 meters long. And you have to pack it in this small rounded structure known as the nuclei. Okay, so it's almost like taking a rope from right here, from the University of Jordan, all the way to the airport. So we're talking about 35 kilometers, and you have to wrap it and pack it and put it inside a yellow tennis ball. It's quite amazing. How can you pack this large molecule into this small structure? Now, something else. If you take all of the DNA molecules of an adult individual, just like we're talking about one single individual, you take all the DNA molecules in all of his cells or her cells, and you tie them to each other, you would go to the sun and come back like 500 times, something like that, just from one single individual. Quite amazing. So here what I'm talking about is how can we pack this DNA molecule into this small structure? This takes place via proteins known as histones. So what happens here is that the DNA molecule is wrapped around histones, and then these histones get packed together, okay, into a really compact structure, okay, known as the chromosome. Now, whenever you have a combination of DNA and proteins, like histones, for example, you have a structure that is known as chromatin. So right here in yellow, you have the DNA molecule, and it is wrapped around this blue structure, okay, or known as histones. Okay, now this here is known as a chromatin molecule. So the basic structure of a chromatin molecule is known as a nucleosome. So, okay, so what is a nucleosome now? Okay, so the idea is the following. You have histone molecules. Well, actually, okay, so you have histone molecules, and there are 5 types. Right here, I'm showing just 4. So you have 4 types in here. And these are known as histone 2A, histone 2B, histone 3, histone 4. So there are 4 types of histone molecules, and there are duplicates of each. So you have 2 of histone 2A, 2 of histone 2B, 2 of histone 3, and 2 of histone 4. They form an octameric molecule. Octa means 8. So I have this octameric molecule, and wrapped around this octameric molecule is 146 base pair, 146 base pair DNA molecule. Base pair because you have 146 on one strand and 146 on the other one. So we say base pair. So, okay, so you have this molecule right here. Now, this here is known as the nucleosome. So what a nucleosome is specifically is the following. It's the octameric structure of histones, of the 4 histones, or I should say the 4 types of histones, or 8 subunits. And you have the DNA that is wrapped around it, and you have a free DNA. Okay, this is known as the linker DNA. So this structure right here, the octamer, plus the DNA that is wrapped around it, plus the free DNA is known as the nucleosome. Okay, same thing here. Okay, so that's the octameric molecule, the DNA wrapped around, and you have the histone-free DNA. This is known as a nucleosome. Now, what happens right here is that you have the octameric, again, structure, you have the DNA wrapped around it, and you have another histone. This is the 5th histone, and it's known as histone 1. It's sort of like it seals the interaction between the DNA and the octameric structure. So this here is known as a chromatosome. Okay, so basically if you have the octameric molecule right here plus the free DNA, and you can have histone H1 involved, this is known as a nucleosome. Okay, but if you look at the structure only this here, right here, without the linker DNA, then it is known as a chromatosome. Okay, so different terms, but they mean different things. So this is another view for the different structures. So the yellow circles represents the histone octamer. Okay, so the H2, H2A, H2B, H3, H4, and 2 of each. Now here you have the nucleosome core, where basically you have the octamer plus the DNA that is wrapped around it. Here you have the chromatosome, so you have the nucleosome core plus the histone 1 protein. And here you have the nucleosome, where you have the chromatosome, the octamer, the DNA wrapped around it. You can have the histone 1, and you have the linker DNA right here. Okay, now some books represent the linker DNA as half of this DNA and half on the other side. Okay, like in here again. So here you have the octamer, the nucleosome core particle. You have the octamer plus the DNA that is wrapped around it. Here you have the chromatosome having the nucleosome core particle plus part of you have the histone H1, and here you have the linker DNA with the chromatosome. Okay, now again, the nucleosome may or may not have the histone H1. Now, the thing is with histones is that they bind to DNA forming a chromatin. Again, a chromatin is a combination of DNA and proteins, specifically histones, and the DNA is packed. Okay, so it keeps on coiling, and it keeps on looping and so on, forming a structure, a compact structure of DNA. Now, if we have the DNA being packed and highly compact, it is known as a heterochromatin. Okay? If you have the DNA loose, as what you can see in here, if you have this loose structure, then it is known as a euchromatin. Now, the difference between a euchromatin and a heterochromatin, or the conversion of a euchromatin into a heterochromatin or heterochromatin to a euchromatin, is really dependent on histone H1. What histone H1 does is that it really packs the DNA molecule. If it is released from the chromosome, if it is released from the DNA becomes a loose structure. Again, this has important functional implications when we talk about transcription. Okay, so someone would say, خلاص, I'm fed up with all of these terminologies. So what is DNA? What is chromosome? What is chromatin? How about chromatid? What's a gene? We'll talk about gene later on. Okay, I know a lot of terms, but let's look at it this way. So chrome, by the way, means color. Okay, zome means object or body. If I say chromosome, I'm talking about a colored body. The reason is that when it was first discovered in the late 1800s, they were able to color this structure, chromosomes, and they said, oh, that's the colored body. This is chromosome. And from this term, they came up with the different terms like chromatin and chromatid and so on. Okay, so let's look at the different terms right here. Okay, so that's a chromosome. So it is a complete structure of DNA molecule. Okay, a single unit. Now, in our cells, usually we have only one arm, and this arm is known as a chromatid. Now, when cells want to undergo meiosis, they duplicate this arm into another arm, and it's known as a sister chromatid. So one arm is known as a chromatid, and you have another arm, and it's known as a chromatid, or specifically sister chromatid. Whether it is just one chromatid or having a sister chromatid associated with it, it's known as a chromosome. Okay, now if you look at this, if you look at the chromosome structure and you zoom in, you would find that it is basically a complex of two things, DNA and protein. So it's known as a chromatin. So you zoom in and you're not looking at chromosome anymore, or you're not looking at chromatid anymore. You're rather looking at a DNA molecule plus proteins associated with it, with the DNA molecule. Now, if it is highly packed, it is a heterochromatin. If it is loosely packed, it is a euchromatin. Now, you look at just a single molecule, or you zoom in further, and you notice the octameric structure plus the free linker DNA. It links the different histones to each other, and it is known as a nucleosome. So this structure right here is known as a nucleosome. So if you look at this structure only, the octamer plus the DNA that is wrapped around it, you know it is a chromatosome. So let's release the histone molecules, and you would only get a DNA molecule, which is a double-stranded helical molecule. Zoom in further, and you will notice that each one of these strands is mainly composed of nucleotides. Okay, and these are the T, A, C, T, G, whatever, and they are and the two of them are complementary to each other, and they are anti-parallel. Okay, now a certain sequence of nucleotides within the DNA molecule would make up the code for a gene. So right here you have a certain sequence of nucleotides, and they make up a gene right here. And you have another sequence, and it codes for another gene. And here you have a region that is intergenic, that is a region that is between genes. Okay, so you have maybe a third region right here, and it encodes another gene. Now please note the terms that I'm using. So it encodes, because here you have a code right here, like a computer code. Okay, so you have a code. So this sequence encodes another gene, and so on. So a gene is located on the DNA molecule, and the DNA molecule is associated with histones forming nucleosomes, and you have nucleosomes wrapped around each other forming a chromatin structure, and this chromatin structure is part of a chromosome. And the chromosome can be made of just one chromatid or two chromatids. Okay, a chromosome and its sister chromatid. Okay, now remember something else. This lecture is full of terms. Now remember something else. We are diploid. Our cells are diploid. What do we mean by that? It means that if you have a cell that has only one chromosome, it means we say it's a haploid. Okay, I'm not talking about chromatids, I'm just talking about full chromosome here. Now our cells are diploid, meaning that we have two copies of every chromosome. One chromosome comes from the mother and the other chromosome comes from the father. Okay, so these two chromosomes, we call them homologous chromosomes. Homologous chromosomes. Now, if I have this as an X structure, okay, if I have this as an X structure, it means it is just, again, it's same thing. It's a haploid, but it's composed of two chromatids, okay, sister chromatids. Now, so these two are homologous chromosomes, like chromosome 1 from chromosome 1. You have one coming from the father and you have the other coming from the mother, and so on. So you have chromosome 2. You can also have it diploid. Two copies of chromosome 2, one from the father, the other from the mother, and so on. And this takes place, by the way, in meiosis, right? So you have two homologous chromosomes, and in meiosis what happens is that each one of them would go into a different cell. So here you have a haploid cell. This is a diploid cell. Okay, now this is also a haploid cell, just like this one, except that here you have the chromosomes formed made of sister chromatids, but here you have chromosomes made of just single chromatids. I hope that it's clear now. Okay, so finally we'll talk about, briefly we'll talk about RNA molecules. So RNA molecules, basically they consist of long, again, unbranched, there are no branches, just single strand, chain of nucleotides joined by phosphodiester bonds, just like DNA molecules, between the 3' end of the hydroxyl group of 1 pentose and the 5' phosphate group of the next. Now the pentose is a ribose, not a deoxyribose. The pyrimidine bases include uracil and cytosine, not thymine, just like DNA. Now, usually in our cells, RNA is a single-stranded molecule. Okay, DNA is double-stranded. Okay, there are always exceptions, all the time. Now, the thing about DNA is that it has a specific double helical structure, right? But for RNA, it does not. It's a single strand, and it can form different structures, by the way. And within these structures, you can have the formation of hydrogen bonds. When can you have this within an RNA molecule? Whenever you have the bases complementary to each other. Yes, they can form, they can loop around, and they can form hydrogen bonds among themselves. Okay, so different RNA molecules can have different three-dimensional structures. So RNA is different than DNA in that regard. Now, you've probably learned that there are three types of RNA molecules, and these are the messenger RNA, which is not written here in this table, amazingly. And you have tRNA or transfer RNA, and you have the ribosomal RNA. So these are the classical RNA molecules. But scientists have discovered a large number of different RNA molecules of different structures and different sequences, and they have specific structures. The ones that I marked here with a star are the ones that we will talk about in this course. So tRNA and RNA molecules are involved in translation, messenger RNA translation. Now, microRNA molecules, or miRNA molecules, are involved in post-transcriptional gene expression. So we'll talk about these later on. You have short interfering RNA, or RNA. They are involved in RNA interference. They have similar structure, similar function as microRNA molecules, interfering with the function of messenger RNA in using it for translation, using it for protein synthesis. And we have what is known as long non-coding RNA molecules, and these are involved in regulation of gene expression. Now, you're lucky that you don't have to know all of the functions of the different RNA molecules, because they are really important. They haven't discovered, most of them have been discovered in the past 5-6 years. So we don't know much about what they do exactly, but they do a lot of important things. Okay, so I know it's a long lecture, a lot of terminologies. Pay attention to the wordings that I said, and the terms, the definitions of the different terms. We will meet and discuss this lecture later on. Allah ma'akum.